Okay, so today we're going to finish up talking about chapter 5 in terms of the hemoglobin myoglobin material. Again, this won't be on your exam Wednesday and or Friday if there's another snow day, but it may be on either the next exam or it'll definitely be on the next quiz. So last time we left off, this material actually is going to be covered on the exam number 2. We talked about the transfer of oxygen from the lungs in hemoglobin to the tissues, all the way to transfer through myoglobin, and how carbonic anhydrase generates protons, which result in a lowered pH. The lower pH promote those ion-ion interactions and stabilize the T state or deoxygenated hemoglobin. At the same time that this occurs, the carbon dioxide modifies the deoxyhemoglobin to form this carbon dioxide carbamate at the N terminus of hemoglobin. This carbamate ion then travels to the red blood cells, to the lungs, and we're able to release carbon dioxide. So this is available on As You Learn, but here it is also just kind of step by step what's happening in regards to the transfer of both carbon dioxide and oxygen in respiration. Okay, so thinking about this clicker question. What is the effect of high pH? So high pH is low, so if we think about high pH, we have low protons, so low proton concentration, oops. If we have a low proton concentration, that means those ion-ion interactions are destabilized. If the ion-ion interactions are destabilized, that means we're forming the R state and we have a increased affinity for oxygen. All right, so I don't have the stylus because I'm working from home, but I can highlight everything in red that I'm going to write. How about that? Okay, and so if we think about that, these are the types of logic, or the type of logic that you should use in tomorrow's, Wednesday's exam in order to kind of think about what would happen in different events or in different interactions with different heterotrophic or homotrophic modulators. Okay, so the answer should be, C, right? Increases the affinity for oxygen because the R state is stabilized based off the reasoning that I showed here. All right, so we've talked a lot about cooperativity in regards to hemoglobin. And so thus far we have talked about cooperative binding. So cooperativity exists in a group of proteins known as allosteric proteins. And again, this is not just found in hemoglobin. Hemoglobin is just kind of our generic protein of interest, but there's lots and lots of different allosteric proteins. And we'll see this again in glycolysis. Glycolysis uses allosteric proteins for regulation. So we'll see this a lot in metabolism, and specifically the metabolism cycle that we're going to talk about is glycolysis. So I'd like to introduce it now, and then we'll again talk about it in chapter 14 and 15 when we talk about metabolism. So what are allosteric proteins? Essentially, allosteric proteins are when you have more than one binding site on your protein. And so the binding of a ligand to your protein at a site other than your active site or ligand protein binding site. All right. They also induce a conformational change in that protein. And so those are the two kind of properties that allosteric proteins have. One, they have a site other than their active site where a heterotrophic modulator or effector will bind. Again, this can either activate or inhibit your enzyme activity. All right, and so it changes your enzyme activity. You have the ability to change the ability of more than one molecule to bind, and you induce always a conformational change in the protein. All right, so those are allosteric proteins. So let's look again at one other heterotrophic modulator, this 2,3-bisphosphoglycerate. This is the molecule here, 2,3-BPG. Again, we'll see this in glycolysis, but it actually plays a role in hemoglobin oxygen binding as well. So this is an example of another or additional heterotrophic modulator that affects oxygen binding in hemoglobin. There we go. And so what this small molecule does is it actually binds right in the center cavity of hemoglobin. So here's our hemoglobin quaternary structure where we have the 4 subunits, and 2,3-BPG or 2,3-bisphosphoglycerate binds right in the central cavity of the T state. All right, so it binds tightly to deoxy. It stabilizes the T state because it keeps this cavity open, keeps the central cavity open, and therefore promotes the ion-ion pairing and stabilizes the T state. All right. If the T state is stabilized, again, we're going to reduce our affinity for oxygen. And so our allosteric binding curve will actually be shifted to the right. All right. And the levels of BPG, this is a figure from your textbook. All it's representing really is that the levels of BPG stay or vary at different altitudes. And in fact, our blood always under homeostatic or homeostasis conditions always has BPG present in it. Otherwise, our binding curve would look more similar to hemoglobin than, or sorry, more similar to myoglobin than hemoglobin. And so this is just showing that the presence of bisphosphoglycerate, 2,3-bisphosphoglycerate, actually shifts the affinity to the right and therefore lowers the oxygen affinity of hemoglobin. Okay, so looking at this question, in the curve below, the binding of oxygen to hemoglobin is given in curve number 3, shown here. Select the answer that gives the curve number representing the addition of 2,3-bisphosphoglycerate. So we've already said that 2,3-BPG binds at the central cavity. Therefore, it stabilizes the T state and lowers the affinity of oxygen. All right. We know that when our affinity lowers on our curves, that our curve shifts to the right. And so the answer would be, the answer would actually be curve number 4, right? So the answer would be D. All right. So that's the last heterotrophic modulator that I want to talk about. Here's kind of a summary. The 3 heterotrophic modulators that we talked about, or effectors, as they result in hemoglobin binding and conformation or affinity for oxygen, either increasing or decreasing, are proton, carbon dioxide, and now 2,3-bisphosphoglycerate. Okay. So that summarizes chapter 5, the chapter 5 material. Okay. So moving into chapter 6, we're going to build on all the structure and functional work that we've done in regards to proteins, and we're going to build on that and talk about enzymes. And again, I like to split this chapter up into 2 parts. Part 1 is just talking about kind of the properties of enzymes in general, and part 2 is talking about the kinetics of enzymes in regards to Michaelis-Menten kinetics and inhibition. All right. So this is kind of a general scheme of enzymes required to, there are 2 conditions for life. All right. Organisms must replicate and they must be able to catalyze chemical reactions, both efficiently and selectively. These are both Very important terms. We're going to talk about why enzymes are so important in regards to this condition for life for number two, because they are able to catalyze these chemical reactions in a selective and efficient manner. Down here, we're going to see lots of these in this chapter 6, part 1. This is known as a reaction coordinate diagram. The coordinates are, you have free energy here, or delta G. Remember, we've already talked about free energy. That tells you whether a reaction is spontaneous or not. All right, and then so that's on your y-axis. Your x-axis is literally just the progress of the reaction. And so we start with an enzyme and a substrate. We undergo some activation barrier, and we're left with an enzyme and a product that is formed. All right, so why do we require enzymes instead of just a regular chemical catalyst? And so obviously chemical catalysts are prevalent in our society. They're used for processes like natural gas conversion to methanol, all sorts of different things, generating fertilizer, lots and lots of different processes. But our body, instead of using chemical catalysts, we use enzymes, and we do this for these 4 reasons. The first are that the reaction rates for catalytic reactions converted by enzymes are actually much higher in the presence of an enzyme than uncatalyzed. So the first thing I'll say now is that all the reactions that occur by an enzyme will occur naturally, just at a much, much lower rate. So that's what this table from your textbook is showing. This is actually table 11-1 from your textbook that's showing the, this might have been from our old textbook, sorry. That's showing the progress of chemical reactions. And so here you can see that we've talked about carbonic anhydrase, which converts carbon dioxide right here. Carbon dioxide to bicarbonate and protons. That's carbonic anhydrase. That reaction will occur at a very slow rate, 0.13 times per second in the absence. So just in buffer, that reaction will occur. When the enzyme carbonic anhydrase is generated, that's increased 7.7 times 10 to the 6 times. And so that makes it a viable enzymatic reaction in order to affect hemoglobin and oxygen binding. Okay, so I like this graph because it just demonstrates the difference between the rate of reactions between non-enzyme reactions and in the presence of an enzyme. Unlike chemical catalysts, these reactions all occur at physiological pH and under physiological conditions. So many chemical catalysts require high temperatures, high pressures, basic or acidic conditions. Here we have essentially physiological temperatures, atmospheric pressure, and neutral pHs. So they're very, very mild in the reactions in the conditions that are required to undergo the catalysis. They are very specific. I just said that. That means there are very few side products, and so the specificity of each reaction is very, very high. And we'll talk more about this again in chapter 14 and 15 when we talk about metabolism. But these reactions are extremely highly regulated. And we'll talk about 3 different ways that enzymes are regulated. One we've just talked about, right, in chapter 5. They undergo this allosteric control with the heterotrophic modulators in order to either increase or decrease their reaction that they catalyze. They can also be covalently modified. Usually a phosphate group is added. And we're going to talk more about free energy in terms of delta G and relating that to equilibrium. And so we're going to see that the enzyme concentration can be varied, and that concentration variability is really plays a nice role in regulation of enzymatic reactions. Okay, so now let's get into a little bit of chapter 6.1. So what are enzymes? Enzymes are really just proteins or biomolecules. So biomolecules are proteins that can do chemistry. That's the very basic, easy definition of what an enzyme is. All right, so biomolecules are proteins that catalyze a specific chemical reaction. How do they do that? They do this by lowering the activation energy. All right, and that's what we're going to talk about in class on Friday, is really what the activation energy is. But essentially, we've all learned this in intro chemistry. The activation energy is represented here on the reaction coordinate diagram, and it's represented by delta G double dagger. And I'll go more into that in class because I don't know where my double dagger symbol is on Friday. Okay, so they lower the activation energy. That's represented here. Delta G. I will try to insert the double dagger. Let's see. Symbol. Just a bunch of emojis. Don't want you guys to get confused. I don't know why. Here we go. Pictograms. It looks like two kind of, you guys can look it up. I honestly don't see it. Double dagger, double dagger. Technical symbols. Sorry. Oh my gosh. All right, well, I guess PowerPoint doesn't have it. I just don't want you to get confused in the difference between free energy, which is relating entropy and enthalpy, to the activation energy, which is delta G double dagger. So we'll talk more about that on Friday in class, just because I'm not able to see the symbol. Yeah, sorry. Okay. So delta G, I guess I'll just write double dagger. There you go. And that's represented by this large increase in free energy. So that's the amount of free energy needed in order to complete the chemical reaction. So the majority of proteins, and all the proteins that we're going to talk about this semester, are comprised of proteins. So they are made up of our 20 amino acids. There are, however, just like the ribosome, proteins or enzymes, that are comprised of RNA molecules, with the most popular always being referred to as the ribosome. And so the ribosome obviously is the organelle that is involved in protein synthesis, and this very important enzyme is comprised of predominantly of RNA molecules. Ribosomal RNA makes peptide bonds during translation. Hopefully you've talked a lot about that mechanism that occurs in other classes. It's a beautiful mechanism that we used to talk about, but we just don't have time to talk about that in this course. All right, so the majority of the enzymes, or all of the enzymes we're going to talk about this semester, are actually proteins, okay? So what happens, what are enzymes? They help the system reach equilibrium faster, but this is important. They do not change the equilibrium of the reaction. They can, that means that they don't affect the delta G, that they just, Aiden, I'm working. Jesus. Sorry. This helps the system reach equilibrium quicker. And they can work in the forward and reverse direction. So it's very, very important that you realize that enzymes do not determine the delta G. That means they do not determine whether a reaction is spontaneous or not. Right? Enzymes do not determine that. This is very, very important. They are just going to speed up the rate of the reaction, not whether it will occur or not. That is determined by the free energy in regards to, so far what we've learned, delta G equals delta H minus T delta S, right? Again, here's a reaction coordinate. We have our delta G. We'll talk a little bit about how to look at this on Friday and determine whether the reaction is endothermic or exothermic. But you can see here the reaction and process without the enzyme in black and with the enzyme in red does not affect the overall delta G of the reaction. All right, this is actually an exothermic reaction. It's negative, so it's favorable overall, and the overall delta G is unaffected by the presence of enzyme. The only thing that is affected is the activation energy required for the reaction to proceed. And so it's lowered, so those reaction rates are increased dramatically. Okay, so just some terms that we want to throw around here are enzymatic cofactors. Enzymatic cofactors are just small biomolecules that are needed for the reaction, for the chemical reaction to occur. They can be biomolecules or any small compound. Chemical compound. All right. And so they are a chemical that's required for activity to occur. Okay, so here's just some notes from last semester. So a chemical reaction that occurs, remember that's the activity of your enzyme. We talked about that when we talked about assays for detecting proteins. We talked about colorimetric assays or assays to determine quantitation or the amount. And we also talked about assays to determine the activity. And now we're going to define activity as a measurement of whether the chemical reaction occurred or not. So cofactors, within cofactors we have different subsets. The first is a metal ion that we'll talk about when we talk about mechanism. The second is a coenzyme. A coenzyme is, instead of a metal ion, it's an organic molecule that's required for activity. We've already talked about one coenzyme in regards to heme, which is a prosthetic group. We talked about this both in hemoglobin and myoglobin. A prosthetic group, remember, is permanently affixed to your protein. Oops. And it's required for chemical catalysis to occur. All right, the second are these co-substrates. And we're going to talk about two co-substrates mostly, this NAD plus and NADH, which those of you in lab have seen, and this flavin mononucleotide, both in the oxidized and reduced state. And we'll talk more about these in length in chapter 13, metabolism. But I'd just like you to to be introduced now to cofactors so that in chapter 13, you kind of know why they're important and why we're talking about them. Okay, we need to talk about two forms of your enzymes, both APO and HOLO. APO is the form of your enzyme in the absence of any cofactors. Remember that your enzymatic cofactors are required for activity, right here, required for activity. And so in the absence of any cofactors that are required for activity, you'll have inactive protein, which means it will not be able to do the chemistry that it's supposed to be. This inactivated form of your protein is termed APO. When it's in the presence of its cofactor and you have an enzyme cofactor complex, that term is called HOLO. So the protein is whole, it's active, you have all the cofactors necessary, and it's doing the chemistry that you want. So make sure we'll use these terms APO versus HOLO throughout the semester, and make sure you are familiar with the difference in the two of them. Okay, so why do enzymes really require cofactors? The predominant reason is because proteins cannot go oxidation reduction or group transfer reactions very easily. All right, so this is really the reason. It's easy for enzymes, because of their amino acids and their pKa's, to participate in acid-base reactions, or to have a charge-charge interaction, or to form a covalent bond for the reasons we've talked about. We know all about pKa's. The pKa can affect whether an amino acid is an acid or a base, or whether it's protonated or deprotonated, and can have charge-charge interactions. Or in the case of a disulfide bond, distance can determine whether a disulfide bond will be present and form a covalent bond or not. But if we're doing electron transfer reactions or the transfer of a functional group like a phosphate group, we need to have cofactors present in order for this reaction to occur. Okay? Again, we're going to talk about this again in chapter 13, particularly in assisting in redox chemistry or the electron transfer. This is the functionality that's occurring in the reduction of NAD+. All right, so this is the moiety of the nicotinamide, which is what the N stands for. So nicotinamide adenine dinucleotide. This is just the nicotinamide functional group associated with that chemical reaction. So we can add a hydride molecule and gain 2 electrons, and we can generate NADH. We have an oxidized form here, is equal to NADH, or sorry, NAD+, and the reduced is equal to NADH. And it's reduced because if you remember oxidation reduction chemistry, you're gaining electrons. When you gain electrons, you're more reduced. All right, so we can use these cofactors in these oxidation reduction reactions. And right now, you're not going to have to identify what's being oxidized and reduced. That'll be more for chapter 13. But I just want you to see and visualize like this example, alcohol dehydrogenase, the conversion or the oxidation of ethanol to acetaldehyde is in concert with the reduction of NAD+ to NADH. Remember, you always have to have reduction and oxidation occurring at the same time. Okay, so just a few more things. Let's talk a little bit about classifications of enzymes. And so enzymes are always named with ACE at the end of them. There's a very complicated nomenclature that is involved in naming enzymes, especially new ones. And there's a especially numbering system that your textbook goes about explaining in detail. I don't think it's very important. What I think is important is that you understand, we add ACE to the end of the enzymes when naming them, and that the beginning usually describes an enzyme's substrate or the catalytic action that it's performing. All right, so there's 6 different classifications of enzymes. I want to make sure that you know all 6 of these. We'll do a couple of examples, but this will be on your next quiz and on your homework assignment that's due, that I'll try to assign this week. It'll be due next week, and you'll have to be able to classify chemical reactions visually by looking by each of the 6 classifications. All right, so the first we just talked about, oxidoreductase or oxidation reduction. These are redox reactions. They're involved in chemical transfer, and they require cofactors. So if you see the presence of those 2 types of cofactors present, NADH, FMN, there's a few more that we'll talk about as well, that automatically signals to you that an oxidoreductase is occurring. Transferases are transfer of a functional group. We'll see these a lot in phosphoryl group transfer, so ATP transfer. That's kind of the go-to transfer group that we'll talk about this semester, especially in regards to glycolysis and gluconeogenesis. All right, but any transfer of a functional group can be occurred. This is just what we'll talk about mostly. Hydrolysis reaction is the addition of water, so adding water to break bonds. So this is actually the reverse reaction of what we've talked about all semester in regards to dehydration reaction, where we've removed water. Now the hydrolysis reaction is actually adding water in order to break bonds, not make them. Lyase reactions are elimination of a functional group in order to form a double bond. You're usually cleaving a carbon-carbon, carbon-oxygen, or carbon-nitrogen bond. Isomerase is just an isomerization reaction. So the number of atoms will remain the same. Oops. But the arrangement is different. All right. And then a ligase is actually a ligase is actually the formation of a carbon-carbon, carbon-sulfur, carbon-oxygen, or carbon-nitrogen bond by a condensation reaction. And it's usually coupled to the cleavage of an ATP molecule. All right, so let's work a couple of examples like you may see on your homework or in a future quiz. So the first example is showing this reaction here, where you have pyruvate being transferred or being converted to oxaloacetate by the enzyme pyruvate carboxylase. Okay, and you may get confused and see that it's the ATP is being hydrolyzed to ADP and phosphate, and so you may want to characterize this as a transferase, but it's not, because you're not transferring the functional group, this phosphate group, to your substrate to make product, all right? But what's happening is, shown in red, you have a carbon dioxide that's actually forming a carbon-carbon double bond, shown here, coupled to the hydrolysis of ATP. So this would be defined as a ligase. So new carbon-carbon carbon-carbon bond is formed during the conversion. ATP is hydrolyzed. Here's the new bond, and so we would refer to this as a ligase. Okay. So this would be a clicker in class. What class does this enzyme belong to? And so now we're looking again in red. We have a phosphate group here on 1,3-bisphosphoglycerate. This reaction is converting 1,3-BPG to 3-PG, all right, 3-phosphoglycerate. This is found in glycolysis by the enzyme phosphoglycerate kinase, a very important enzyme that we'll talk about. Now when we look at red, something's happening to this phosphate group. It's actually being transferred from the substrate 1,3-BPG to ADP to make ATP, all right? So we're transferring a phosphoryl group. I guess that's 2 R's. From 1,3-bisphosphoglycerate to ATP, okay? Now because that transfer is occurring from our reactant, we can classify this now as a transferase, okay? So let me insert a shape. So the answer will be transferase, because it's a group transfer reaction. Sorry. Okay. All right. So the last example that I want to talk about today is this classification, where we're converting glucose 6-phosphate here to fructose 6-phosphate, which is another sugar. And we'll again see this. All of these are glycolytic enzymes. So this is preparing us for our metabolism portion of the semester. And so we're converting glucose 6-phosphate to fructose 6-phosphate. These have, if you sat and counted the number of atoms, both of these have the same number of atoms. They are just rearranged differently. And so you would hopefully, based off of our identification of the different classifications, be able to classify this reaction as an isomerase. Oh, great. Which is not one of the answers. Transferase, ligase, hydrolase. Goody. That's a win. I must have changed the question. So this is an isomerization reaction. None of the above. Goody. Fail. Oops. All right. So that concludes today's lecture. Again, this will be on a quiz over the next, probably next Monday. We'll have a quiz depending on how far in the material we get to. Okay. Have a nice day.